Hello there everyone, and welcome to this Biology Mind A-Level tutorial on functions of polysaccharides. So just to recap, in the last lesson we learned that polysaccharides are formed through condensation reactions and are broken down via hydrolysis reactions. You now know that there are three types of polysaccharides: starch, glycogen and cellulose. So in this tutorial, we're going to have a closer look at these three types of polysaccharides, so starch, then glycogen and cellulose. So let's get started without further ado. So we've already talked about what the polysaccharides are that we're going to explore in this tutorial. Now, why don't you have a go at brainstorming some different functions of polysaccharides before we go into it? So pause the video at this point, and I'm about to reveal the next slide. So, starch, let's start off with starch first of all. So, starch is the key energy store in plants, and excess glucose is stored in this form. So, starch is essentially, it's the storage polysaccharide of plant cells. So, it's the polysaccharide used for storage of glucose. We store excess glucose in the form of starch. Now, there are two polysaccharides that form starch. Number one is amylose and number two is amylopectin. So we're going to look at those in the next couple of slides. For now, what I want you to appreciate is starch is the energy store in plants. So it's stored in plant cells and it's insoluble, which is a really valuable feature of it because it means that it doesn't lower the osmotic potential of inside the cell versus outside the cell. So it prevents osmotic effects such as swelling or bursting of cells by water moving in or out, which is a really great property of starch. So now let's have a look at its two major polysaccharide components. So amylose is one of them. And amylose has a structure adapted for compact storage. And the reason it's adapted for compact storage is because it's made up of these alpha-glucose monosaccharides joined together in condensation reactions, as we've met before, joined together to form this long polysaccharide chain of amylose, which in turn is coiled, which means that it has a really nice compact structure, has a helical structure, a bit like DNA, so it really fits in really compactly into the plant cell, which is a really valuable property of amylose. Now, the reason why it forms these straight chains that are then coiled is because amylose has something that we call 1,4 glycosidic bonds. Now, all this means is it forms glycosidic bonds, which remember are formed in the process of condensation, the condensation reaction. So it forms these bonds between carbons 1 and 4 of the alpha-glucose molecules. So let's just draw this out just to make it a little bit clearer for you. So let's say this is our first glucose. To draw these hexagonal shapes. So there we go. Now, let's have a think about which parts of this are our glycosidic bonds. So, we know that our glycosidic bonds are these bits here, in between are each individual glucose monosaccharide. Now remember, these are alpha-glucose monosaccharides. And the reason why we call it 1,4 glycosidic bonds is it's formed between carbons 1 and 4 of the structure. So, if we label this one up as well, you can see that the glycosidic bond is formed between carbon 1 of one monosaccharide, so of one alpha-glucose molecule, and carbon 4 of the next alpha-glucose molecule. So it forms these lovely straight chains. So this becomes relevant in a second when we go on to talk about amylopectin. So amylopectin has a structure adapted for fast breakdown. Now, the reason why it's adapted for fast breakdown is because it's got this branch structure that you can see over here. So it has this backbone of alpha glucose monosaccharides, and coming off it are more chains of alpha glucose monosaccharides. So we call this a branch structure. It's not just alpha glucose arranged in a single line, it's alpha glucose monosaccharides branching off other chains of alpha glucose monosaccharides. So the reason why this happens is because we say that amylopectin has not only 1 4 glycosidic bonds, but also 1 6 glycosidic bonds. So it forms bonds also at the carbon 6 position. So I'll try to draw that out again for you as clearly as I can. So this is our glycosidic bond in carbon 4 position. Okay, so we know that that was carbon 4 position, so this is carbon 1, this is carbon 6 up here, this is 5, this is 4, this is 3, this is 2, this is 1. So these are our glycosidic bonds as normal between carbons 1 and 4 of each alpha glucose monosaccharide. But there's also a bond between carbons 1 and 6. So you can imagine if there's further alpha glucose molecules here in this chain coming off from here in this 1 4 position, so it's just coming off in this straight line, and there's also other 1 4 glycosidic bonds in this chain here, each of these individual chains can link up also by the 1 6 glycosidic bond, which is why you get these straight chains of where alpha glucose is bonded in the 1 4 glycosidic bond position. But then you also get these branches coming off where at this point, for example, this alpha glucose, its carbon number 1 is bonded to the carbon number 6 via this glycosidic bond, so hence forms a branch point. So I thought I'd explain that in a little bit more detail than necessary, just so that you understand why amylose is not branched and amylopectin is branched. So as a result of amylopectin being branched, the reason why this allows for fast breakdown of amylopectin is because, let's say our enzyme here is in, let's put it in purple. So let's say our amylase enzyme here comes along and wants to break down our amylopectin. It has more points at which to work at because our amylopectin is lovely and branched. So that means that amylopectin has a structure adapted to fast breakdown. It has this increased surface area for enzymes to work on because of the branching. Okay, so that's amylopectin. So remember, starch is formed of the 2 polysaccharides, amylose and amylopectin. Amylose is not branched, amylopectin is branched, which means that amylopectin allows for nice fast breakdown of starch, whereas amylose allows for nice compact storage of starch within the plant cell. Now, we're going to contrast this all to glycogen. Now, unlike starch, glycogen is the storage polysaccharide of animal cells. Now remember I said that starch was the storage polysaccharide of plant cells. So in the same way, glycogen is the storage polysaccharide of animal cells. And it has a structure adapted for storage and fast breakdown. So it's adapted to its storage function and has more branches than amylopectin, making it easier to break down. So you can see it's also a branched structure, very much like amylopectin. The only difference is it has more branches than amylopectin. Now, In terms of its similarities to starch, and really to amylose and amylopectin, is it's formed of these alpha glucose monosaccharides as well. So glycogen is stored in liver cells, and its breakdown is controlled by the hypothalamus with the use of endocrine hormones. In particular, we're talking about glucagon. So we're going to think more about this over the next few slides. Before we move on, something that I'd just like to point out is, why do you think animals have glycogen but plants don't? Because as we can see, glycogen is even better adapted than amylopectin for fast breakdown. So why don't plants need that? Have a think about it. Okay, so the reason why plants don't require glycogen but animal cells do is because animals in general are much more mobile than plants. They have a higher metabolic requirement than plant cells. So therefore they rely on this fast breakdown nature of glycogen, whereas plant cells can have the slower but still fast breakdown of amylopectin with its less branched structure, because plant cells tend to have a lower metabolic requirements. That's a nice little fun fact for you, and that also can sort of help you remember that glycogen, since it's in animal cells, is the one that is more branched and therefore the one that breaks down faster, whereas amylopectin is that storage polysaccharide that is less branched and still adapted to be a good surface for enzymes to work on and to break down fast, but not as fast as glycogen can release its monosaccharides. So just to reiterate my point, amylopectin and glycogen are both branched. Now, glycogen is much more branched, whereas amylopectin is branched less than glycogen. However, in terms of their similarities, they are both made of alpha glucose chains. Alright, so we're going to have a look at this in a little bit more context now. So Sarah has just eaten a very large meal. So look at all the glucose, look at all the carbohydrates contained in this meal. So we know that carbohydrates are broken down into simple sugars when we eat, when we digest carbohydrates, and they're broken down into their simple monosaccharide form of glucose. So there's now a high concentration of glucose in the blood. And this is detected by cells of the pancreas. And what happens is the beta cells of the pancreas secrete insulin. And insulin in turn is going to bind to its receptor, as you can see here. Now this is going to stimulate various changes in the cell, including the movement of glucose transport channels to the surface of the cell, and also it causes these glucose channels to work more efficiently. So insulin has many roles in cells, and it causes more efficient uptake of glucose in many different ways. But overall, I think the most important concept for you to remember is that it allows for glucose to be taken up into the cell and out of the blood. So our glucose molecules now move into our cell, therefore reducing our glucose concentration in our blood, and also allowing for any excess glucose that's not used in respiration to be stored as glycogen. Okay, and the main glycogen stores are in the liver and in the muscle. Okay, now Richard has just done an intense gym session. So the opposite is going to happen. He's going to have really low glucose in his blood. And now the alpha cells of the pancreas are going to secrete glucagon. And glucagon is going to bind to glucagon is going to stimulate breakdown of glycogen into glucose, which means that glucose can then pass into the blood and be utilised by cells in respiration. And the glucose concentration in the blood rises back to normal. So that was just the way in which glycogen is very easily utilised in terms of breaking it down and adding to it in the animal body. Okay, so now that we've talked about glycogen, let's think about cellulose. So we're going back to plants here. So cellulose has a structure adapted to being strong. So the chains of cellulose form these tough microfibrils, which give it strength. So in contrast to glycogen and to starch, cellulose is made of beta-glucose monosaccharides. So that's one contrast to think about. And it's arranged in these linear structures to form chains of cellulose. And it forms these tough microfibrils which give plant cell walls really good structural support, which is one of the reasons why when we draw plant cells, we tend to draw them as these rectangular structures. Whereas when we draw animal cells, we draw them as more of like a sort of a spherical shape, don't we, with a few little inundations in it. And that's because animal cell membranes are formed of this very fluid phospholipid bilayer, whereas plant cell walls are formed from this strong cellulose, which gives it really good structure. Okay, so we've gone through cellulose, starch, and glycogen. So this is a really nice summary table to summarise everything that we've talked about. I'd really recommend you jot it down in your notes because it has lots of really key information. So let's just go through it bit by bit just to recap. So cellulose is found in plant cells. Its monosaccharide unit is beta-glucose. And we know that it forms these straight chains. Therefore, remember, it has these 1,4 glycosidic bonds, which I explained earlier in the tutorial. And it's not branched because it just has these 1,4 glycosidic bonds between carbon 1 of one monosaccharide and carbon 4 of the next monosaccharide. Now, starch is formed from the polysaccharides amylopectin and amylose. They're both found in plants. They both have the same monosaccharide unit of alpha glucose. The difference is that amylopectin is branched. Sorry, this table is slightly wrong. So amylopectin is branched. So it has bonds at 1,4, carbon 1 and 4 position, and 1,6 position. So they've got this slightly the wrong way. Oh well, it's a learning curve. Whereas, which means that because amylopectin has bonds at 1,4 and 1,6, it's a branch structure, which means it has this structure. Now, in contrast, please mind the scribbles, but this is now correct. So amylase has glycosidic bonds in the 1,4 position, which means that it's not branched. So it forms this nice coiled helical structure, whereas this is branched, and we know that glycogen is also branched. And now glycogen, like starch, is also formed from alpha glucose monosaccharides, and it's branched because it's got glycosidic bonds at the 1,4 and 1,6 carbon positions. And it's the difference between amylopectin and glycogen is that glycogen is much more branched, so it tends to have branches every 10 alpha glucose monosaccharides, whereas amylopectin tends to have branches every 20 alpha glucose monosaccharides. And remember, glycogen is found in animal cells. Okay, so that was a really chunky tutorial there. If any of that doesn't make sense, please go back and watch my explanation again, and I hope that this will make your learning of polysaccharides a little bit easier. Well done for following through till the end, and I'll see you for the next tutorial.